Howdy, folks. Thanks for checking in to Mr. Ulrich's LandofBiology.com. I again am Mr. Ulrich. In this kind of third, maybe addendum to our series on photosynthesis, we're going to be talking about alternative photosynthetic pathways. We've really been focusing on what's called the C3 pathway, so I'm going to introduce the C4 and the CAM pathways. To start comparing these different photosynthetic pathways, it's probably good for us to start at just what are the resources that plants need in order to go through photosynthesis, the different chemical reactions of photosynthesis in the first place. So for the light reactions, of course, they're going to need light, which is most often going to come from the sun, and they're going to need water, and most of the time that's going to come from the ground. For the Calvin cycle, they're going to need the products of the light reaction, of course, but they're also going to need dioxide, which is available in ever increasing amounts from the atmosphere. So plants have evolved different adaptations, different structures in order to help them obtain these resources. Like leaves act as solar collectors, but since that's where photosynthesis, the splitting of water is going to take place, that's where the water is required. And the problem, of course, is that the water comes into the plant through the roots. And so in order to get the water up through the roots and out up to the leaves, it has to lose water. So we got some transpiration that's going to take place, and that's going to happen because there are little openings in the leaf for evaporation to take place and for gas exchange for photosynthesis. So we'll talk a little bit about stomates later on. But there's also stems in order to get the water from the roots up to the leaves, at least when we're talking about your standard tracheophyte. Let's take a quick look at the cross-section of a typical C3 leaf. Here we got the palisades layer stacked up on top of the spongy layer. Together they make the mesophyll. And this is where most of the photosynthesis takes place. And the gaps there between the different cells down in the spongy layer are actually air spaces. To keep them air, we have the epidermis, the upper epidermis there, and the waxy cuticle that keeps the water from the rain from soaking into those air spaces and making them water spaces instead of air spaces. Down below, we have the stomates we were talking about before. Each stomate is made of 2 guard cells. As the stomates open, they allow for water to evaporate. As the water evaporates, that allows for the replacement of the water from the vascular bundle, which we'll talk about in a second. But that allows for replacement from below and down from the roots, and we end up with transpiration taking place. But that's also going to allow for the escape of oxygen that's produced through the light cycle and allow for the taking in of carbon dioxide for the Calvin cycle. So that gas exchange takes place through the stomate. In the vein of the cell, in the vascular bundle, we got the xylem that provides the water from the roots, and the phloem, which transports sugar around where it needs to go. Together, they make the vascular bundle. Certainly, the amount of water that's available for a plant cell is going to be important to its ability to go through photosynthesis in the first place. So hot, dry days can pose some problems. See, stomates have to close in order to conserve water. And the way the stomates work, remember there are 2 guard cells, and as the guard cells take on water, they plump up, and that causes them to elongate, and they kind of open, they kind of pucker as they get longer. And so now the stomate is open as there's plenty of water available. As they lose water, they shrink up, and that causes them to close. So this clearly is an adaptation to living on land because we're talking about water evaporating, but it's going to pose a couple problems. One of the problems are those pesky stomates and the way they work. What happens with the stomate, of course, is as the water evaporates from the airspace, that allows more water to move up the xylem and is replaced into the cells themselves. And so water actually, in order for it to work, a plant has to lose water in order to gain water. But thankfully, the stomate's open, and that allows transpiration to take place. The plant is also going to need some carbon dioxide, and there are 2 sources of carbon dioxide. One's going to be the atmosphere, of course. Stomate's open, allows for gas exchange. We get some carbon dioxide. Another source is going to be the cells themselves because they're going through cellular respiration. So they're going to be producing some carbon dioxide of their own. But as they go through those light reactions, they're going to be producing lots of oxygen. Most of that is going to be gotten rid of through, again, gas exchange via the stomate. But those cells themselves are going through aerobic respiration. So some of those cells are going to use the oxygen that is produced. So we have a little gas exchange right within the cell or between the cells. And we also have some gas exchange with the environment. But the problem is those stomates are going to close. When they close, that stops the evaporation of water, which might sound good for the conservation of water, but it's also bad because it shuts off transpiration, and now no more water is going to be moving up into the leaf itself. It's also going to stop gas exchange. It's going to allow oxygen to build up from those light reactions. It's also going to allow the amount of carbon dioxide to go down as the Calvin cycle goes through, and this is going to lead to some problems. This is going to lead us directly to our next problem, which is with good old Rubisco, ribulose bisphosphate carboxylase. And I know I spoke so highly of it in the last video. It is that all-important enzyme in the Calvin cycle that's involved in carbon fixation. It takes the carbon and carbon dioxide and actually bonds it to the ribulose bisphosphate. It does this because it has an active site that bonds with carbon dioxide very nicely, and in doing so, it reduces the Ruby P, and we build some sugars. This actually puts the synthesis in photosynthesis. Now this works only when there's a lot of carbon dioxide and not a whole lot of oxygen around. When we reverse that and we have a whole lot of oxygen around, well, now the Rubisco is going to bond the oxygen to the Ruby P. Because oxygen actually fits in that active site in Rubisco just like carbon dioxide does. And what this does is it causes the oxidation of Ruby P and is going to lead to basically the breakdown of Ruby P. That's causing the breakdown of sugars. This is a process we call photorespiration because it uses up oxygen. When carbon dioxide is available, good old Rubisco is going to go through the Calvin cycle that we've been talking about and produce that 3-carbon sugar, glyceraldehyde 3-phosphate. Now, this is why we call it a C3 plant, because that's a 3-carbon sugar. So carbon dioxide makes a 3-carbon sugar when it's fixed. When there's a lot of oxygen around, that Rubisco bonds with the oxygen and instead breaks the Ruby P down into 3-carbon and 2-carbon sugars, which make their way to the mitochondrion, where they're lost as carbon dioxide. The problem is they never make ATP. So this photorespiration is a way to lose oxygen. Over the course of the last billion years, the amount of oxygen present in the environment has been quite variable, and this has had quite an effect on the types of photosynthesis and the types of metabolic pathways that were crafted by evolution through those periods of time. Having stomates is all well and good if you're a terrestrial organism. If you're an algae that leads an aqueous existence, you have the problem of carbon dioxide not diffusing very readily through the water, and so they have problems with carbon dioxide supply. Many of them have evolved these unique structures called pyrenoids, which are actually carbon concentrating structures. What they do is they take carbon dioxide from the water and they make it into a storable compound, which they can then convert back into carbon dioxide as the algae needs it. Essentially, photorespiration is the oxidation of ribulose bisphosphate. It causes a short circuiting of the Calvin cycle because the carbons go to carbon dioxide rather than to glucose. And in fact, about half of the carbons that are fixed through the Calvin cycle can turn around and be lost through photorespiration. And this obviously is going to reduce the efficiency of photosynthesis because we're not making any ATP, you're not making any glucose through this process. And if we could just get photorespiration to not take place as often, well, then we can make the plant a heck of a lot more efficient. And so this kind of push towards efficiency for the plant creates a strong selective pressure towards some other way that doesn't cause the amount of oxygen to build up that it does. Then plants can achieve a higher photosynthetic efficiency if they just could reduce photorespiration. And some plants do this. What they do is they separate carbon fixation from the Calvin cycle. One group of plants are called C4 plants. What they do is they actually physically separate those two processes in two different types of cells. They have one type of cell where this carbon fixation takes place and another type of cell where the Calvin cycle takes place. So first what they're doing is they're taking the carbon and they are fixing it into 4-carbon structures. That's why they're called C4 plants, because carbon is fixed into a 4-carbon compound rather than in a C3 plant where it's fixed into a 3-carbon G3P compound. Now, in order to do this, they're going to use a different enzyme. So now we don't have to worry about competition with oxygen because we're using a different enzyme, PEP carboxylase, rather than Rubisco. It's also going to require a different leaf structure so we can achieve that compartmentalization, those different types of cells. Another type of plant are called CAM plants. These separate carbon fixation from the Calvin cycle temporally. So we separate it. Well, we don't, but the CAM plants do. They fix carbon during the night and store that in 4-carbon compounds, and then go through the Calvin cycle during the day, slowly peeling those carbons off of the 4-carbon compounds to run their Calvin cycle while the stomates are closed. We're going to see this in succulents like cactus and bromeliads like pineapple. C4 plants have come up with a better way to hold on to carbon dioxide. So taking a first step before the Calvin cycle takes place to fix that carbon dioxide with PEP carboxylase as that 4-carbon compound. This allows us to keep those stomates closed so that they can retain water during the day and then open it up and allow transpiration to take place at night when it's not so bad and they don't have to worry about losing so much moisture. This again is going to require a different leaf anatomy so that we can deal with the stomates opening and closing at night as well as water supply. The types of plants we see this in are grasses and sugarcane and corn. And if you think about it, these are adapted for hot, dry, open, blowy plains types of places. Let's take a look at the cross-section of a C4 leaf. When we look at the cross-section of a C3 leaf, we see that, like I said, palisade layer, the spongy mesophyll with the air spaces. When we look at the C4 leaf, I got that vascular bundle, and then surrounding the vascular bundle is a layer of bundle sheath cells. And then surrounding the bundle sheath cells are some mesophyll cells. It's in the mesophyll that the light reaction is going to take place, so that's going to produce some oxygen. The oxygen is going to diffuse into the air spaces there between the cells of the mesophyll. And so when the stomates are open, the oxygen can leave and we can take in some carbon dioxide. That carbon dioxide goes into the mesophyll cells and is fixed by PEP carboxylase into oxaloacetate. That oxaloacetate is turned into malate and is stored as a 4-carbon sugar so that in the bundle sheath cell where the Calvin cycle takes place, that's where the Rubisco hangs out, it can peel off 1 carbon at a time and go through and make some sugars. Now, we're keeping the Rubisco separate from the oxygen. I don't have to worry about PEP carboxylase getting in touch with the oxygen because it has a much higher affinity for carbon dioxide than it has for oxygen. So it doesn't have the same problem that Rubisco has. It's going to fix that carbon, like we said, into that 4-carbon compound, which it can store for later. Taking a real quick side-by-side look at the two plants. Here the C3 plant, you can see the spongy mesophyll and even the palisade layer. A lot of those cells have direct contact with the external environment via the stomate. We're over here in the C4 plant. Those bundle sheath cells are surrounded by the mesophyll cells, and that's where we find the Rubisco. It is separate from the one. So we have a physical compartmentalization, so we keep the oxygen away from the cells where they're going through, from the Rubisco, where they're going through the Calvin cycle. All this reduction in photorespiration makes C4 plants pretty efficient and pretty successful. They make up only about 5% of the plant biomass, but they do about 30% of terrestrial carbon fixation, so that's pretty darn successful. Now, the deal is that they use more ATP through their metabolic pathways than the C3 plants do. And that's actually part of their claim to fame. It's part of what makes it possible for them to do what they do. We've talked about the C3 plants. We talked about the C4 plants. Now we need to talk about the CAM plants. CAM stands for crassulacean acid metabolism. And like C4, this is also an adaptation for hot and dry climate. Where the C4 plants separate carbon fixation and Calvin cycle by location within the leaf, these guys actually separate them by time. Now, normally we think of, in a normal plant, the stomates being open during the day. That's going to allow for the oxygen that's being produced through photolysis to escape and not build up in the tissues. We know that can cause a problem with rubisco. Because it's open during the day, we're also going to allow for a lot of evapotranspiration to take place, which is going to keep water moving up the plant. Good thing. And it also provides a time for carbon dioxide to make its way into the leaf. The problem in a hot and dry climate is that if you open up your stomates during the day, evapotranspiration can take place really quickly and kind of too much and eventually dry the plant out. And so what these CAM plants do is they close their stomates during the day. But they still need to exchange gases, so they open them up at night. Now, the problem here is that since the time of high carbon dioxide use is going to be during the daytime, when the products of the light reaction are readily available for the Calvin cycle, we're going to need some carbon dioxide during the daytime as well. And if we close up our stomates, well, then there's not going to be carbon dioxide during the day. So what these CAM plants do is they save up the carbon dioxide. They store it up kind of like the pyrenoids do. What these guys do is they store up the carbon dioxide by fixing it together into 4 carbon storage compounds. And then as the day goes on, they can kind of peel off the carbon dioxide one at a time from these storage compounds and keep the concentration of carbon dioxide high enough in the cells to prevent the oxidation of rubisco. We see this in all kinds of succulents and some cacti, but it's a lot of desert plants that we see this in. Desert plants and, oh yeah, pineapple, which we typically don't think of as desert so much, but they do kind of like the dry environment. To wrap this up, looking at C4 and CAM comparatively, both of them are trying to solve this oppositional problem of having your stomates open for gas exchange, but you're going to lose too much water if you do that. The C4 plants are going to do that through location anatomically by having different types of cells, where the CAM plants are going to do this temporally. They're going to do this at different times. What exactly was the problem with the C3 pathway in the first place, right? Why do we need all of these alternative pathways? Well, when we start talking about need and development and stuff like this, we're really getting into the evolution. We got to remember that rubisco probably is a progenitor of the oxygen revolution, so it was evolving when there was a high carbon dioxide, low oxygen environment. That low oxygen environment did not really provide the selective pressure against an active site that can also accommodate oxygen because it just wasn't around. Nowadays, things are a little bit different. Plenty of oxygen in the air, thankfully, and not very much carbon dioxide, although that amount is certainly increasing. With all this oxygen, that means that on a typical hot, dry day, a plant can lose about half of the carbon that was fixed through Calvin cycle just through photorespiration and lose that. And this inefficiency really presents a pretty strong selective pressure for the development of a different pathway. Well, I certainly think that's enough for now. Thanks again for checking into Mr. Ulrich's landofbiology.com. I am Mr. Ulrich, and if you have any questions, feel free to drop me an email, write something in the comments section below. Other than that, we'll see you in class.